Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. This week, I'm with Rodrigo Santos, the Chief Technology Officer at clock.bio, a company who are doing incredible work in cellular rejuvenation, particularly focused on age-related diseases. Rodrigo and his team have taken a hypothesis-free approach to their research, which has led to some unique insights. But we also talked to Rodrigo about his journey from a small farming village in Portugal to tackling rejuvenation through his obsession with Yamanaka factors. We talked about the lessons he's learned about balancing science and business, and also why emphasis on execution is key. Well, I'm back on Careers in Discovery and very excited to be joined by Rodrigo Santos of clock.bio. Rodrigo, it's been a long time coming, but welcome to the show.
Thank you for having me, Tom.
Of course. Good to see you. So, Rodrigo, I want to talk a little bit about what you're up to at the moment. Obviously, recently joined clock.bio as the CTO. Tell me a bit about what you're up to. Tell me a bit about decoding rejuvenation.
Pleasure. Yeah, so thank you, Tom. Yeah, my name is Rodrigo. I'm joined clock.bio in July this year, and I'm CTO there. The company's been around now for 2 years, but I only joined in July.
Yeah.
And at clock.bio, we work on this health spam aging field, and our mission is all about finding these novel therapeutic pathways to treat age-related disease, to increase health span in line with growing longevity. And we believe that we have a strong scientific and comprehensive approach to tackle this. We are hypothesis agnostic. So we started from an hypothesis-free method to identify all the rejuvenation programs and rejuvenation factors that one could manipulate to increase rejuvenation. And that approach of being completely hypothesis-free was very interesting, and that's what the main reason that attracted me to join them back in July.
Yeah. My understanding is the kind of principle is, look, some cells don't age, so let's figure out why those cells don't age and how we can apply those lessons. Is that, I appreciate that's probably an oversimplification, but is that...
It's not an oversimplification. Right? It's actually... that's the kudos, and that's really the ethos of how the company started. Many of our co-founders, they have this background in stem cell research. So they've been growing stem cells, especially industry-potent stem cells and embryonic stem cells for many years. And these cells, they can be kept in culture forever.
Right.
And that really triggered this thinking, as you know, from this from looking about aging, that stem cells don't age. It's one of the features of these cells. They really keep these hallmarks of aging at the bay. So we thought, well, that's a very interesting approach. So is that a way we could force them to age, especially in a transient fashion? So Kobe Brands, a postdoc in Marcotta's lab, did try that experiment during his postdoc. And they tried a couple of different mechanisms, and they found aging stimuli that you could, in a transient way, express in these pluripotent stem cells, in these pluripotent stem cells. And 7 days after these stimuli, and looking at them phenotypically, these stem cells were showing markers of aging stem cells.
Okay.
They were showing mitochondrial dysfunction by reduced oxygen consumption rate. They were showing some DNA damage. And this was very interesting because these were all the hallmarks that somatic cells in adult individuals show and are part of these hallmarks of aging. This 12 hallmarks wheel that is becoming super famous nowadays. But even more interesting was because that was a transient stimuli that you could control when it was present and absent. After the removal of the stimuli and analyzing these cells again in 2 weeks' time, they were again indistinguishable from the original material.
Interesting.
So, meaning they rejuvenated or they de-aged, as you prefer to call them.
Yes.
And so in 3 weeks, we had this full circle going from non-age to back to rejuvenated. So, which was in our mind a very interesting aging model in a dish, where we can now ask very comprehensive, unbiased questions.
Yes.
And that's when the company started 2 years ago. And what they were thinking at the time was, could we go and identify the impact of every gene in this rejuvenation and in this aging? And how do you do that? Nowadays, you do that by functional genomics. So it's how we go about it. So they embarked in a big and costly project that just finished, and we disclose it now in September or October, that we call Atlas of Rejuvenation Factors. So what we did, we used single cell CRISPR screens. So we managed to knock out every single gene that was expressed in these iPS cells, and that's around 13,000. Right. And also to activate the remaining genes that were not expressed, so that's around 7,000. So at the end of this, we run this in 3 different buckets of 2 knockout screens and 1 activation screen. We look at the impact of this removal or activation in what we call aging signature. So we derive a transcriptomic aging signature, where we could see what are the genes that should be expressed or not expressed in aging iPS cell or in a non-age iPS cell. And we come up with a ranked list of around 150 genes whose manipulation affect this aging and rejuvenation of the stem cells. Okay. So we were quite happy with it, because it was a quite concise list of genes that some of them were previously identified in aging. So we were some positive controls there. Other genes were in pathways that make sense, and we were quite happy about it. And some of these genes are actually in pathways that we were not aware of, that would have an impact in aging. So quite new targets. Some of these genes work in a way that if you inhibit them, they promote rejuvenation. So that's exciting, because it's an easier therapeutic methodology, because we could go out with small molecules and inhibit this. Yes. So we now are at this phase where we have a ranked list of these 150 targets that have this impact, and we are quite ready for this next stage to really bring these targets that we identify in a complete unbiased screen and show if they work in adult tissue.
Interesting. And there's so many questions I have about this, Rodrigo, but I'm going to be selective. I suppose using this hypothesis-free approach is interesting in itself, in that in some ways it allows you more freedom. In some ways, I guess it requires a bit more discipline as well, actually, because you have to, at some point, generate a hypothesis, right? That must have been a very, in some ways, refreshing, in some ways, frustrating place to start from.
I'm a technologist by heart, and so I love that side of being hypothesis free. That really drove me. And up to this date, what Clock set up to do was actually impossible, because many of these aging models out there, they are more comprehensive, but they don't allow such a unbiased screen.
Yes.
Some of them, they just take too long. So let's look at aging in a mouse model. Okay, that's it's more as comprehensive as it could be in a non-human rodent model. But still, how are you going to run this complete unbiased perturbation approach in an animal model? Is impossible.
Yes.
And so here in 3 weeks, we could get this information, and then you can take it as you want. Okay, it's criticizable because it's quite a synthetic model, and a lot of this is reduced to a transcriptomic signature. Absolutely. But now we have hypotheses, and you have these targets that we can now, it's a defined list that now we can take and validate somewhere else. And what we like about it is that we've been doing a lot of bioinformatics work on and of course a lot of literature work on these targets, and they are highly present in disease. Right. So we've been clustering and do these target disease association studies by bringing data from different public available data sets and looking at what pathways they are involved. And these are all pathways that are triggers of the age-related diseases, such as muscle loss, such as neurodegeneration. So initially, one could say, oh, you're looking for factors that the only thing they do is to contribute to the aging of the stem cells in this particular stimuli. but actually, we come up because we were looking into this from a transcriptomic perspective and look at all these aging hallmarks. The list of genes we have, they are quite pan-aging and quite observable in disease. So how do you go from here, hypothesis-driven, to biotech, which is a hypothesis-free to an hypothesis-driven approach? And that is actually now the big shift that the company is now going through, and it's a super exciting approach of, okay, now that we go from here, how do you create a therapeutic product? And again, as I was saying, we're trying to be led by the science, and at the moment the science is telling us that there is 2 or 3 disease indications, therapeutic areas that it makes more sense to go, and again, based from the targets that we are identifying, and even from there we're trying to generate our assays in a way that they will give us as more data as possible, in a way that is a digital record that we can feed back to an analysis tool that will enable a lab in a loop, the experimental design. But it's one of the biggest step changes for our company, because they all started from a perspective of, okay, let's just keep this going in an unbiased way for long as we can. Almost, as you could say, from a technology perspective, and now we're moving more into a scientific perspective. Okay, let's work backwards now. So now that we have disorders and you have pathways and you have targets, and we want to make sure that we'll have the easiest therapeutic modality possible, how does this all marry? So we can narrow down that list of 150 targets to our top 5. So we can start our drug discovery campaigns to have drug candidates based on these top 5 targets that make sense from druggability, from disease, from pathways, from toxicology. Are these targets that are new? Are these heavy hitters that maybe some molecules have been identified, but they have to be optimized? And all that thinking is the current thinking that Clock is going through.
Yeah, it makes sense. And this is perhaps not on the same scale that you're talking about here, but this is a common shift that a lot of biotech companies go through in terms of mindset and culture, right? You go from this big experimental freedom where anything goes, anything's possible, let's figure out what's going on, to, okay, now we need to develop a product and we need to focus and we need to have more discipline in what we pay attention to. And
and we will try to keep going for long as it makes sense. To really keep this platform and this data generation for because there's still a lot of impact out there. So we now run this screen and these iPSCs, and we have the biggest data set out there. There's 20 terabytes of data where we have a matrix of every single knockout or activation, and then have this read by single cell, where we now have the transcriptome of the impact of every gene deletion or every gene activation in pluripotent stem cells. And this is a fantastic data set for anything you can think of. One can now think of, is there a possibility to create an in silico aging cell? Can we now really think about AI and how do you go about and completely manipulate aging in virtually? And now we could, because now we have this super perturbation data set where you can see the impact of every gene on this aging outcome, but also in every gene expression on the cell. So you're now starting bringing more data to this, and you can start thinking of maybe you have this possibility of making complete in silico predictions that will tailor your drug discovery. So that bit will continue, because data generation and then using them, there is a big win on it. But of course, going down we at the end we want to be a biotech. We're a biotech we'll be developing therapeutic products. So we cannot fall in love with a platform and forget about the real goal, which is to really have an impact on health span and develop products. So this new thinking, and that's what we are doing now with the current seed stage. It's really develop these plans and start inroads into defining these targets. What are them? How do you prioritize them? How do they correlate to disease? And start drug discovery campaigns, hopefully in collaboration with pharma partners.
I'm sure there'll be a lot of interest because it's a very attractive concept that, if these stem cells can de-age or rejuvenate, theoretically, does that allow us to question the inevitability of aging? That's an attractive question to ask, right? That's a very complex and exciting question to be exploring.
The feedback we've been getting from a
Mm-hmm.
lot of the pharma discussions we're having goes that way. From and we're quite excited with some early discussions with around 10 pharma companies going at the moment, and they are very interesting.
Yeah.
And a lot of these farmers are actually starting to think, what is the next big thing? Some of them managed to embark on obesity, some haven't, and they are thinking, okay, if we miss obesity, what is next?
Right.
And for sure, a way to look into age-related disorders from the onset is quite appealing, especially if you're coming from perspective of having new targets that will have an impact. So discussions are ongoing, and Clock is doing the groundwork to make sure we have work to show on the validation of these targets and come up with a plan to take this into validation phase.
Yes. Well, it's very exciting, and I could talk about this part of it all day, but we're here to talk about you, Rodrigo. So,
okay.
So yeah, I wanna move on to your career, because I think you've had a very interesting career. You've worked in some very different organizations at different stages. There's been a thread of obviously this idea of functional genomics running through a lot of your career. You started out as a cellular molecular biologist. That was sort of the original training that you had. Tell us a bit about. Let's go back even further than that, though. Tell us a bit about why science, Rodrigo. Why biology? Why this path for you?
Absolutely, and I feel quite honored when I talk about my path, because I like to be reminded that I'm quite happy where I am, and one of the problems of, I guess, the day-to-day job is you forget where how you got there, and really and really makes you sometimes sad, and then you need to take a break to really reframe. And so my parents are farmers, so I came from a 300-person village in rural Portugal. I'm the youngest son of two brothers that couldn't care less about school, and my parents, they both left school after 4 years in school. So in my village, there was only one person that actually ever been to university before me, and so my path was traced. It was all about, okay, you are going to either go in construction or be a farmer. That those are the only two options. My brothers, I had summer jobs with them, working construction, every summer working in farming. I would help my parents over the weekends, and that was it. Actually, it took a lot of convincing from my head teachers before going from primary school, secondary school, to convince my mom to let me go to secondary school, because for her it was all about, it's just a waste of time. It's going to be like his brothers, and it's not happening. So from early age, for some reason, and I still don't know why, I want to be a scientist. And in Portugal, we have these little booklets. Every student have them, even in primary school, where you have the name of your parents. So basically, those booklets is for you to take home with message from the teacher saying, "Oh, your kid was bad behavior, was misbehaving today. So please sign over that you agree that I can take this action." But one of the sections of those booklets was what you want to do when you are old. And I always wrote scientist. I have no idea what the scientist was, but I wanted to explore. And today I'm a scientist, and I'm still a scientist, and that's what drives me. And so that drove me to then eventually get to university. I love biology. I love chemistry. So I went and studied biochemistry. I quite liked that. And then I learned about molecular biology. I quite loved it. This DNA, recombinant DNA technology, I thought this is very interesting. I want to explore this. So I did a master's in recombinant DNA, mainly in plants. From there, I got into a PG program And during that PhD program, I learned about this fundamental study from a Japanese scientist called Yamanaka. And this was 2011. The paper was in 2006, but it took 5 years from that paper being out for me to learn about it. I was not aware that it was possible to reprogram old cells back to pluripotency. And when I saw that, I really almost made it was like a religion. That was a Bible to me. This is what I want to devote the rest of my life. If it's possible to control cell plasticity, this could be the fundamental backbone of all the therapies for the future.
Right.
And if there is a thread that has guided my career since then, is that paper. And is this concept that if you can control cell plasticity, this can really lead to new therapies.
Yes.
And that's been the thread so far that I've been following. I interviewed in several labs in Europe and the US that they work in iPSCs. I decided to come to Cambridge because when I came for the interview, I realized that the Stem Cell Institute was next door to the Judge Business School.
Okay.
And it was interesting because you can go to these classes at Judge Business School for free if you are at Cambridge University. So I decided, okay, I can come and be a 2-in-1. And for several years, I did that. I was doing my PhD 9 to 5, and then in the evening, going to Judge Business School and then some classes in management or finance. In one of these classes, I met Darren Disley, which was the CEO of Horizon Discovery, and I quite liked what they were talking about, this gene engineering. And then suddenly, CRISPR was published,
right?
And then there was this press release that Horizon was licensing CRISPR technology, and I thought, actually, now that we can control cell plasticity from iPS cells and we can do gene engineering from CRISPR, this is a 2-in-1. So now you can. And I decided to apply to Horizon, and that was my first job. And I quite liked it there. Did a lot of gene engineering in stem cells. By doing this, then this academic in Cambridge University, Mark Cotter, came up with this system to make cell differentiation quicker based on genome engineering. BitBio was born, and I joined them. It was very, very interesting there to really bring, again, this cell plasticity, but now from a cell therapy perspective, can you do this quicker? And from this, then I joined Mogify. Again, same concept. Can we now control cell plasticity, but instead of indirectly in the lab and then lead for transplantation, can you do it directly in vivo? And that is a super holy grail in therapy. And it was quite a bold mission, so I joined them. I was with them for 4 years, and quite interesting project that we work on. So I was in these 3 jobs between around 10 years. And I thought maybe it's time for me to see not just operational and the management and kind of the technical side, but more how do we take it from the other side, company creation. And that's when I joined Novo Holdings, and I worked with them for 2 years as an entrepreneur in residence in company creation, mainly exploring gene and cell therapy and then RNA therapeutics. And we created a couple of companies in this RNA space. And suddenly this opportunity at Clock appeared, and same concept. How can we control cellular plasticity, in this case, not to convert cells from one to the other, but how to convert from old cell to a young cell? Again, same concept. It's the same cell, we just modulate the plasticity of the cell, in this case for the aging or rejuvenation. And I found it very, very interesting again. And
so this discovery of the Yamanaka factors has really been the core of what you've been working on. What do you think? You said that as soon as you sort of came across this research that it captured your attention. What do you think it was that caught your eye about this? I mean, obviously it's influenced a lot of people, so it's an influential piece anyway, but
for me, what biology, the simplicity of biology sometimes is beautiful. And I think the reason why I fell in love with biology the first time was the fact that everything is controlled by 4 bases. And you're having A, G, T, C, 4 different chemistries, and you control life in a completely digital fashion. So now groups of 3 make codons, and now you have these 64 possibilities to make proteins. And it's so smart and completely programmable. And although biology is not physics where there is law and everything is logical, the more we study it, the more we come up to see that there is actually some governing laws.
Right.
And that paper really showed that again.
Right.
It was so beautiful, so smart, the most simple experiment possible. Like, first experiment was done as I like to conduct experiments, complete black box thinking. The scientist, Takahashi, he just went on these embryonic stem cells, saw which transcription factors are more expressed in here. There's 24 of them. Great. I don't need to think about which ones I will go and test. I will just test them all. Right. And then he did this experiment the same way as James Dyson when he designed the vacuum cleaner who do it. Like, let's just test them all and take one at a time until I come up with the minimal set. And that concept is such a beautiful way. It's just an engineering mindset to biology, eventually settling on 4 factors that on their own control complete this development. We can now revert all the way back. And that was so fundamental and so interesting that, okay, here's a new law that no one knew until now. And it was found in the simplest way. That's why I like it a lot. And made me think, if these 4 factors make this, what are the other programs out there?
Right.
And in the end, is what clock.bio is about. If there is these 4 factors that control going all the way back to pluripotent stem cells, what other pathways control rejuvenation? Maybe there is something that is actually written in our genome, and there are pathways that can be manipulated for this, and that's what we set up to do. What are these rejuvenation pathways that now one can manipulate? And that's what I like a lot about that paper, was the fact that it reduced a very complex problem into an engineering method for really coming down to this set. And then such a beautiful, again, number 4. There's 4 factors that control this. And that day I just, this is incredible.
Yeah, finding the right levers, and you can change everything about life, right? It's an
Correct.
Amazing concept.
Same with CRISPR. Again, CRISPR really caught my attention because it was, again, so beautiful, and so predictable, and again, programmable, and all those concepts of programming biology really excite me.
And this is a difficult question to answer, I suppose, and so I apologize in advance. But what do you see as the sort of extent of the potential of that? I mean, are we going to get to a point where we have complete control over biology, or is it too complex for us to get to? Like, what's your? I appreciate this is very hypothetical, but what's your view on that?
Biology is hyper complex, and there is a lot of heterogeneity, and if you talk about human therapeutics, the beauty about mankind is that we're all different,
right?
Which is good, because if we're all exactly the same genetic trait, every time there was a pandemic, we'll all be dead.
Yes.
So it's good that there is some variability. If not, the human species would not survive, and so that makes this biology with a new level of complexity. And if you look at the genome, there's a lot of redundancy. There is a lot of levers that control the same, but still, we are starting to see a shift on how we interpret complexity. Look at CAR-T, right? The complete programming biology. And if you look into these CARs, in this or T cell receptor T cells, and then this CAR-T T cells, it's all about giving functions to cells that they didn't have it.
Right.
So you program them to do something new, and that, of course, it was for cancer because there was of course, there's always problems and toxicity, and then testing these new technologies. But you can start thinking about, is there a component of really hack biology to work in your favor? And CAR-Ts were the first example of that. So now if you have a complete synthetic chimeric receptor, which is just an amalgamation of components, circuits that you took from other bits that you now collated into a chimeric receptor, and now redesign it to attack cells that were not being seen, and then will trigger an amplification signal that will kill that cancer. It's amazing. In a way, it's a mini robot. It's a mini biological robot, and that you hacked. And this was the beginning. And I do believe that there will be more and more of these applied to a lot of disorders from it was cancer in this case, but there's no reason to believe that this will not be the same for any disorder. And cellular plasticity will have a big role into this. Because up until, let's say, 10 years ago, we did think that cells were completely defined states into these Waddington mountains and valleys. So each cell was defined. The more technologies are developed, single cell technologies, single cell epigenome technologies, you really see that there is a continuum where cell fluctuates between states, old and young, disease, non-disease, behaving more like killer cells, behaving more like suppressed cells. So you see that a cell itself it's not a state that you can now say this is all there is. No, it actually fluctuates, and it depends a lot on the inputs. So if you now can start think about ways to either modify the inputs or modify the way they identify these inputs, you can control this plasticity. And if you shift the needle from a more disease state into a healthy state, or from a young state to an old state to a young state, you can now maybe start crossing these borders of where we are in terms of age, in terms of disease, in terms of any control you want to go. And I do feel this, you start seeing more and more of these hacks in biology and bring them back. The more you understand them, the more then we can modify them.
Yeah. So the limit is really just our understanding of the system at this point, and we're making progress on that all the time.
And this is probably what I see AI having a more impact of really making these completely hypothesis-free ideas, where there's just so much computation power a human brain has, and the way science has evolved, it's been very hard for us to, once we make our careers, to really think broader. We kind of are asked to be thinking smaller about our own gene or our own pathway. And AI could have this power to really just come back and say, okay, actually, if I look at the overall picture, here's this.
Yes.
Of course, the big challenge will be for a scientist to believe it, because it will be so off track that, well, this is just hallucination from this computer. And it might be, but it might be a new way to think about a problem and then actually to bring it and hack it and hack the system in the first place. And I feel that a lot of these future therapies will be based on this programming of biology, but especially about this way to hack cellular plasticity, give functions to things that were not there in the first place, or just revive functions, or really just regenerate or replenish by changing where the cell sits on this continuum.
Yeah, I understand. Interesting. So there's lots that we can be doing there, and lots that we can explore. I suppose one of the interesting things about your career, as you described it there, Rodrigo, is that you had this interest in the business part of things from an early stage, while still focusing on very experimental science, very new science, very unexplored areas, very unanswered questions. And I suppose there's an interesting dichotomy there in balancing this very experimental, very research-driven part of your career with the practical application part of it, which is probably kind of mirrored in the work that you did at Horizon. That was a company that took a very new technology and commercialized it very successfully, right? And so that seems a really good fit for you to have landed at early in your career. But how do you think about those two things, that sort of practical aspect of building a biotech business with the very research-driven experimental end of exploring new technologies?
I think that the business side came from my absolute desire to just keep doing things that are worth doing. And I guess that was my kind of upbringing that taught me that. It's kind of, I didn't really have much time in the past to just think of, I will keep doing. It's all about, if you want to get to university, you got to work, you got to make money. And I feel that, and currently I'm actually reading a book called The Innovators, written by the same chap that wrote Big Jobs and Elon Musk biography. It's a great book because it's about the digital revolution. It's about the invention of computers, from Charles Babbage Alan Turing all the way now to Sam Altman. And there is a trait there on not is often is not the person or the guys who come up with the first invention that eventually turn it into an innovation.
Right.
And they're very completely different things. I guess invention is just coming up with these a trinket that you don't really know what to do. To bring this to something unique and critical and something that will have an impact, you need to have a business component to it, and you need to have absolute execution.
Right.
So an invention without execution and a business plan is just a side note on a book. And you see this over and over. This book tells this over and over. Even the chapter where I'm reading now is about this invention of bitmap. So graphic unit interface is about how Xerox parks. There was this super interesting innovation park by Xerox in California that was trying to imitate Bell Labs. So just having a complete research driven unit outside of the big commercial side of AT&T or Xerox. And Xerox parks had this invented bitmap, which basically each pixel on the screen could be executed in Explorer, meaning that now if you have a mouse, you can go and instead of just being MS-DOS or PC-DOS, you now have an interface. And they sit on it for 2 years, they develop it, and they have no clue what to do with it.
Yes.
Until Steve Jobs got to the doors and saw it. And he actually told them, guys, you are nuts. You could be the biggest IT company in the world. You have this and you don't know what to do with it. And then he did it what Steve Jobs does. He just went back to Apple, stopped everything, said, we are working on this, we are going to get this going. And they created Macintosh, the first interactive graphic unit interface. So again, you took this invention now into innovation by having a business plan and full execution. And I really like execution. For me, execution really dictates. So strategy is one thing, but then is whoever gets it executed. And I see that execution is really well done in a private setting rather than in academia. And that's what I see a big difference. And I'm driven by execution, just get things done and really apply this 80-20 rule daily. Okay, let's just keep going. Let's just do something. Let's keep this going, because strategy will eventually just be a PowerPoint. We need to get something done. And that's what, and I learned that the hard way at Horizon Discovery, because I was in the service team. It was it. You get an email saying you have 11 weeks to execute X, Y, and Z, and you have to deliver Y, A, B, C to this client. And that real execution, you have to get it done, was a big shift. And now I've been, even now that I'm not in the service business, more on the product and therapeutics, trying to bring that mindset. Let's just get pace, cadence, get things done.
Yeah. I always think those early experiences in people's career have a real impact on how they view things long term. You can change your mind about things, of course, but the mentality of delivery, the mentality of pace, the mentality of efficiency and focus and things like that, I think a lot of that is often shaped by where you start your career, right?
Yeah, 100% it is. And then sometimes you're asked to give what is your view on X, Y, and Z? And this is a super biased approach. It's not scientific when people ask for advice. It's only my opinion shaped by my own life, by my own experiences. Like, it might not be the case in your own setting, but he is absolutely shaped. I think I really appreciate having that first job at Horizon Discovery in a service team where it was all about customer focus, delivery. And even if it fails, you have to prove that you did your best to fail, because the client is paying this £40,000 for this project anyway. So you cannot just be, well, you know what? The fuck? That's not possible. It's hard work. You do your job and you get this done, or not done, but proven. And you have cadence, you have milestones, and you have reporting. And that really shaped. And now moving back more into therapeutics, where things are a bit more loose, trying to bring that concept, more the CRO concept into this is quite interesting.
Yeah. And I suppose it's that idea of sort of accepting that things will fail, because this is the sort of world we work in, right? But then being really deliberate about, okay, it failed, but here's what we learned from it, and here's the insight that comes from that, and here's how we apply that learning.
Correct. And sometimes it's all you need to move to the next stage is you need a failure to move on.
Yeah.
Because until you fail, you are into this limbo where you don't really know. And because you don't really know, you have two options. You either act on it or you procrastinate. And this procrastination really leads to derailing. And that is a component that sometimes is this active inaction really is disruptive.
Yeah, absolutely. I was interested as well, Rodrigo, about the time that you spent at Novo Holdings, because that must have been interesting. You said you spent a lot of time on company creation and things like that. And I guess, in that environment, you get to see a number of iterations of that and probably greater volume than you might do normally. What were your sort of takeaways from your couple of years as an entrepreneur in residence with them?
I think it was one of the best jobs I ever had in my life.
Right.
I must say. I was remote, so I was still based in Cambridge, and I would commute to Copenhagen every 2 to 3 weeks for 2 to 3 days. And the team there was spectacular. So I was based with a Novo Seeds, seed investments team, they're called nowadays at Novo Holdings. And then, because I was remote and I was an entrepreneur residence, I would sit with a cluster of people they're called by origin, which is basically where the entrepreneur residents sit. And this is a super divergent group of 25 people. We had highly seasoned CEOs from around the world. I can give you an example. John Moraganor, the CEO of Allen Island, actually was one of us. And I got a lot of insights by speaking with him once in a while. It's just incredible how he thinks about, he loves technology companies and these platforms, but then it's all about how are we going to make this a product first?
Right.
Very sharp questions. And there were medicinal chemists, people with aeronate expertise like myself. So it was quite a diverse group. So we could look into a lot of companies that were getting through the door and be quite critical and very quick look at different ins and outs. And I love it. I think in terms of deal flow, I might have seen over 150 decks from these different companies. And that was the trade-off. Okay, I was there to try to build a company. At the same time, you help us reviewing these other companies. So that's always the trade-off of entrepreneur residence. You give one, you take one.
Yeah.
And that exposure to different things really opened a new chapter in my therapeutics thinking. Because a lot of Novo Holdings was all about what's the therapeutic molecule, what's the chemical matter. And that was something that I was not so used to. And now it's a lot in me on all this thinking. I love it. So takeaways, I love Denmark and I love this people, and I love their thinking and the way to operate. Super professional, hardworking, but then also very personal and say, I have my life, I have my kids, I have my family, and this is work and this is life, and being quite divided between those two without many spills. And I think that's a really good shift from sometimes in Cambridge where everything is just one. It's an amalgamation of what is work and what is life, and then the boundaries are not quite there. I like that quite a lot. And then I like this disease focus first. So disease first, and then you work backwards to technology rather than technology first, and then I'll find a problem for it. That was something that came from that. And I learned a lot about business plans and making these investment memos, kind of what is the fundamental disruption that you are bringing, not just the technology, but what are you doing? What is the market you are playing at? And then, of course, I was at Novo Holdings during the fat cow period. I was there for 2 years, and every quarter it was just, because Novo Holdings is the holding side that owns this around 30% shares of Novo Nordisk.
Right.
And because of the way this works, the charitable this non-profitable status of Novo Nordisk Foundation, these dividends that come from Novo Nordisk will then fall into Novo Nordisk Foundation and then go into Novo Holdings. So every quarter is Novo Nordisk is growing at 30% growth. This is like, how do we use this money?
Yes.
How do you think about this? What are the next big challenges? And you see now that around a month ago, they just built the biggest AI supercomputer in Europe, and it's incredible. This now adapting and trying to make Copenhagen this new Kendall Square, and they even have a name for it. They're trying to make this new Kendall Square in Copenhagen. I really like that Novo Holdings and the Novo Nordisk Foundation is trying to invest directly into their ecosystem. So there is this company, and because of the way they have this articles of association, is making the city where they are based in a biotech system, in a biotech ecosystem, all based organically from this pharmaceutical company. I think that is very interesting. I wish that there will be more of that, where the big pharmas in a city will then contribute to the success of the city.
Yeah, that makes sense. Yeah, and I think it's very exciting. I think the rise of that company is fascinating. I remember when I got into the sector nearly 20 years ago now, they were another mid-sized pharma company. There's nothing particularly sort of distinctive about them to anyone else, and kind of to become, I believe, the biggest company in Europe now, not pharma company, but biggest company in Europe is very interesting. I'm sure there's a lot they can share and give back to that community and that ecosystem through that journey too.
And it's incredible that they do, and it's something that is quite Danish about the fact that because Novo, no one can buy them, because now like Novo Nordisk Foundation, Novo Holdings, because they have this higher voting power, the company cannot be taken private.
Right.
And because of that, they have this really long-term thinking about how to operate. It's not just about hitting the quarter revenues and then these 5-year cycles of how do we go and how do we do, if not we are all out, is very interesting, so organically, so beautiful. And it's very hard to implement in other countries because this system doesn't really exist outside of Denmark. This way to just have this foundation sitting on top and then and getting this in as a committee to look forward. But there it works perfect. It's not the only one. There is more of this type of companies in Denmark, but it's beautiful to have worked with them. I saw something completely different, which I will forever be grateful.
Yes. So looking back, Rodrigo, we always try and leave people with a little bit of advice from these conversations, and I'm sure there's plenty they'll take from what we've talked about. But if you were starting over again, I mean, let's say you were on your first day at Horizon Discovery and you bumped into yourself. With the benefit of hindsight, what would be the advice you would give yourself from a career point of view? What thing would you share?
And this one, I think someone only appreciates their first job once they get the second.
Okay,
especially when they get the third. And that's what I would tell myself. I was, after maybe 6 months, 1 year there, I was hypercritical. I'm in the service business. That's so much I can do. It's quite repetitive, and my hands are tied because all I do is execute and deliver and do this on time to these clients. And then I really got anxious. And I would go back and say, actually, Rodrigo, this is probably one of the biggest lessons you will love in 10 years' time.
Right.
Because this is really defining like pace and cadence. Really, like this is how it should be. Like,
yes.
If not, is a charity or or just an academic group. This is not what it is. Like a business is defined by actually then having a business plan and having some profitability, but then actually doing things that are needed for other people, and that's what you are doing here. So I'll really go back and say, Rodrigo, actually be grateful, and this is, you're learning a lot, you're developing a lot, and what you're learning here will be forever engraved in the way you operate. That delivery is very important. I'll definitely be saying that because and I will learn it after getting second job, third job, is when you look back and say, actually, there is good and bad things everywhere. Not just in this, it's in every company. There is a big difference between press releases and reality in every company. Like you see, even when you are on the inside, and then suddenly there's a press release about the company you work on, and sometimes you think, really? And this is everywhere. Even at Novo, it's no different. because every company is an organization is a group of humans, and humans are flawed, and therefore this just happens everywhere. So and I wish I knew that at my first job in the industry.
Yeah, that makes sense. So try and appreciate where you're at, try and appreciate the lessons that you're learning, and understand that the grass isn't always greener, even though it might look it from over here.
Absolutely, because nowadays, because of social media, especially because of LinkedIn, it really makes people anxious that the grass is always greener everywhere. You look at LinkedIn, how many people report unsuccess on LinkedIn? Like, very rare. It's just like every company is the most successful company in the world, and then you are somewhere and you feel, I wish, and then you kind of do that. You know what? And so that anxiety. So I would go back, and nowadays I feel comfortable with that because it's now my fourth or fifth job, and I see it actually just. This happens is just take your time, things grow. Like it's like stock exchange. If you look at them day to day, there is ups and downs. You need to look in 6 months increments and see if where you are at 6 months is better than where you were 6 months ago. Even if in between you went up and down like Bitcoin. Where is the trade-off when you just look in bigger? And I couldn't really see that in my first job, I would say.
No, that makes sense. Well, Rodrigo, it's been fantastic to talk to you. I am really excited about the potential of what you're working on at Clock, and I will be keeping an eye on that. Thank you so much for joining us.
My pleasure.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.